Welcome everyone to this week's edition of the Extracellular Vesicle Club. This event is endorsed by the International Society for Extracellular Vesicles, and we've been holding this meeting for more than 2.5 years every week, almost every week. We actually took a break last week for the ISEV Tech meeting, which was a meeting on technology and methods that went very well. We had well over 200 participants, lots of demonstrations of new and existing technologies in the EV field, and even a comparison of some reference materials across platforms. So stay tuned for more information about that meeting. And also recently held was the French Society for Extracellular Vesicles meeting, which was also a very successful meeting from what we've heard. And we have here today several representatives of the French Society.
We have also a PhD award, and this year in 2022, the award was given to Mathilde Bergamelli, who did her PhD in Toulouse in the Malmö lab, and who's now working on as a postdoc at the Karolinska Institute. And she's going to present her work today, and we will have the question and the answer afterwards. So Mathilde, congratulations again for your award, and we are now, I guess, listening to you.
Thanks, Christian, for the nice introduction. That was a smooth one. So nice to meet you all. Like Christian said, my name is Mathilde Bergamelli, and I was recently awarded the PhD prize from the FSEV that was presented just before. And I will talk a little bit today about what I've been doing, research speaking during my PhD, but has been held under Dr. Cecile Manu supervision. So my subject was about the placental extracellular vesicles, specifically the small one, during congenital infection by Cytomegalovirus, and what are the potential impacts of those sEV in the fetal compartment. So just to start, I'd like to say that I do not have any conflict of interest for this presentation. And then let's dig into science. So for those of you who are not familiar with the placenta organ, it's really essential for pregnancy success. It is an organ that is evolutive. It's not the same at the first trimester and the third trimester. And it's really essential for the interplay between the mother and the embryo, or later on the mother and the fetus, especially for the secretion of a lot of different placental mediators. All this is really necessary and needed for a balance during pregnancy, that it can be disturbed by different diseases that include metabolic disease like diabetes or hypertension, which has been quite studied a lot in the field of placental extracellular vesicles. But also, and that is what interested me during my PhD, during infectious disease. All this can, of course, create unbalanced communication between mother and embryo, and then lead to placental or fetal defects. The disease I was particularly interested in was the congenital infection by Cytomegalovirus. So this virus is virus from the family of the herpes. So it is quite a big virus, from 150 to 200 nanometers. And this virus has the capacity to be latent in human, and then to reactivate later on potentially. Concerning the transmission, it's mainly in biological fluid, so that includes blood, tears, sexual secretion, amniotic fluid. If there is really an active replication of the virus, it can be present in literally every biological fluid. This is a virus that is strictly human. And if you catch it as an immunocompetent adult, then it's going to be quite okay in most cases. You will not have any major symptoms, perhaps flu-like for a few days. But if you're a pregnant woman and that you catch this virus for the first time, so a prima infection, either if you reactivate this virus during pregnancy, then it can lead to major defects, both in the placenta and in the fetal side. More specifically, on the placental side, it can lead to early miscarriages, to placental calcification. All of those are impairing the future communication between the mother and the embryo. And on the fetal side, there's going to be some defects that are more general, like to the liver or calcification into the intestine parts. But most of the outcomes, the pejorative outcomes that are happening are the defects that are touching the brain and the embryo and fetal brain. There can be some major disturbance in the architecture of the brain, but what are the defects that are the most frequent one are neurosensory damages. So if we have some really early infection during pregnancy, we can have some area loss or some cecity that can be diagnosed only after birth. And this is quite an issue when we talk about the clinical care of this infection, because we do not have, as clinician, that much prognostic tool to predict the outcomes of those neurosensory damages. This is really frequent infection in the Western countries. In France, it's about 1 to 1,000 pregnancies that are concerned by HCMV infection. And what is really concerning as a clinician and also triggering as a researcher is that we do not understand the whole broad of the symptoms that are occurring during this infection. That was a little bit of context and virology context, so a little bit far from EVs, but don't worry, we'll dig into it, because what we've been looking forward in this PhD work was to actually understand what was the mediators that are secreted by the placenta during HCMV that could on one side participate in the pathology that we have in the fetal brain, and also that could be secreted in the maternal blood and serve as biomarkers. I know that most of you are EVologists, so you will definitely understand that we went for placental extracellular vesicles, and more specifically the small ones. So the small extracellular vesicles that are secreted by the placenta are found in various body fluids. They are found in the amniotic fluids, so directly, potentially directly through secretion via the chorea and the amnion. They're also found in the fetal blood. So if we look at cord blood, Miranda will show that there is approximately 45% of the sEV that are positive for placental markers. So that's quite a huge amount. And of course, they are found in the maternal blood, where they could potentially be used as biomarkers. What was really interesting work for us is those two papers that I left here. So Delam, Axman, and Ouyang, they described that the small extracellular vesicles in physiology, so there is absolutely no infection in this context, have an antiviral. So they are protective. If you put those placental sEV, physiological one, in contact with cells and that you secondary infect them, there's going to be a protective effect. What we look at in this work was what is actually happening in case of infection, and specifically in the context of congenital infection by Cytomegalovirus. So the two aims, the massive aim of my work was to analyze what was happening, and I divided this work in two parts. The first one was to analyze what is the impact of HCMV infection on the placental sEV secretion and composition. And then in the second time, of course, in a way, those sEV were modified. And so we analyzed those the impact of those modified placental sEV on the fetal brain cells and the fetal cells. To answer this question, I've been working with two placental models that I will briefly present so that you can get a little bit more insight. The first one is a cellular model. So it's the IPEX cells. It's human invasive proliferative cytotrophoblasts. And those cells are permissive to HCMV. So that's the immunofluorescence that you can see on the right. We have some green staining that is corresponding to the viral protein immediate early. So the pattern that we followed for the collection of sEV was the one that is presented here. So we have 24-hour infection and then a collection of media over 48 hours before sEV preparation. And I will just switch to the other model that is complementary to the cellular model because it's still a placental model, but it's an ex vivo one. So in collaboration with the Maternity Hospital of Toulouse and gynecologists there, We had access to first trimester placenta that were coming from voluntary elective pregnancy termination. And we did a manual dissection. So this is the pictures that you have on the right. And we infected or not alpha of the placenta. So every placenta is its own control for those experiments. And then we cultured them for 2 weeks. I think that this is important because very often when we talk about physiological or pathological impact, we can have some short-term impact, especially on the sEV. But this model is complementary to the cellular model because we have a long-term culture that is still really, as you can see here on the immunochemistry, there is really a maintained structure of the placental tissue. Of course, we did check that there was high infectivity, a successful infection by HCMV. So either with the cellular model or with the ex vivo placental one, we had access to media culture, and that's where I purified the sEV from. We chose to go for a differential ultracentrifugation followed by density gradients. We didn't have that much choice. We could not really concentrate the sEV or go for other purification methods because we are working in infectious conditions. So our main objective was really to get rid of every viral particle that we might have in those conditions. Even if HCMV is a little bit larger than the sEV that we looked at, we still did some systematic infectivity tests for each preparation. So you can see here on the immunofluorescence on the top of the screen that if we have some sEV that come from either non-infected cells or infected cells, that is not enough to induce positive marker, positive staining, for immediate early, so the HCMV protein. I did also check that by RT-qPCR for viral proteins. So that was really our first check. And then in collaboration with Gisela D'Angelo, Isle and Grâce Raposo in Curie Institute, we did some transmission electron microscopy and immunoelectron microscopy to check at the same time the structure of our EVs and also the tetraspanin classical markers. So that is the pictures that you have here. We do not have any difference, at least not statistical difference in the structure or the tetraspanin marker for CD81 and CD9 on immunoelectron microscopy. That was really the first step that we needed to do to make sure that everything was going well so that we can actually go a little bit deeper in comparing the sEV that come from infected or non-infected cells. So specifically for the cytotrophoblastic model, so the first one I show you, the infection by HCMV is increasing the number of sEV that is produced per cell. This is also something that was shown in a different model of infection by HCMV on fibroblast cells. There is no difference in the structure of the sEV that come from non-infected or infected cells. They have both cup shape and everything is classical, if I say. Interestingly, when we look at the wide field images from the transmission electron microscopy from the infected condition, we didn't have anything that resembled a viral capsid or geometrical structure. It was hijacked. So we were happy also about that. And then we also calculate and measure the relative size of the sEV. And we showed that the infection by HCMV is diminishing the relative size of the sEV compared to the non-infected one. So as a global result, we can say that the infection by HCMV is modifying the secretion of the sEV. And then we went a little bit deeper in the characterization of those sEV. So this is the result that we had from a surface marker panel. So through the use of the MaxSpecs exosome kit, so the made panel lists of Milteny, specifically for the sEV that come from placental cells. That was interesting for us to pick that that technique because the placenta rate collection is a little bit lower than the cells. We're with human tissue, so we didn't have access to that many cells. We could not replicate it for a lot of samples. So what we see here is that the infection by HCMV is globally increasing. There is a change in the in the marker and it's in the surface marker. So this is a global change. And specifically, we have an increase in two markers that are CD81 and CD326, also known as EVClub. So most of you know CD81 for its role, its enrichment in sEV and in the exosomal pathway. But CD81 is also specifically known as a cofactor for HMV entry into the cells. So HMV is really a virus that is complex, that is using a lot of different internalization process, and there is a lot of different receptors and co-receptor. CD81 is one of them. So that was interesting for us, that this was the only tetraspanin that was changed and that it was in the context of HMV infection. And then we also showed that there was an increase in the EVClub. And this is also a work that is quite interesting by Nagao and all, is that they did some mouse KO of EVClub and there was absolutely no placentation. And this is apparently really a factor that is needed for placental development. But for now on, we do not have that many more info on human biology. That was for the placenta. And we also did some more analysis on the trophoblastic cells. Like I said, we had some higher rate of collection for the sEV in that model. So we were playing a little bit with the different models and the different, more or less sensitive technique that we had. So for the trophoblastic cells, we went for a proteomic analysis. So here is the result of presented in the volcano plots. So first, we were really happy because we had in our panel 94 proteins that were on the top 100 proteins of exocarta. So that was also a validated check. That was good for us. And then what we were interested about was really to compare the non-infected and the infected condition. And that is the result of the volcano plots. So we have an increase in the sEV that come from infected condition. There is an overexpression of 15 cellular proteins. And there is also the presence of 24 viral proteins. And I will detail those results a little bit more. So the first thing that we saw is that the most overexpressed cellular protein was the Taiwan protein. And this is also a protein that is involved in early stages of a HMV entry. So like I say, HMV is really using a different load of receptors. And we find it quite funny, actually, when we had this Taiwan overexpression, because it really makes sense knowing that it is participating in HMV entry. We also did some predictive pathway analysis, so combining all the cellular proteins that were enriched and that were lower expressed in the EVs that come from infected condition compared to the non-infected ones. And what we saw is that we had all these pathways that were either predictively activated or downregulated. And the striking result is that the first one that was theoretically the more activated pathway is the autophagy pathway. So of course, it makes sense when we know that autophagy and sEV production, EV in general, are really linked. But to us, it had also an additional value is that we know that HMV is a virus that is manipulating autophagy. And this is the results of a French team in Paris. So this virus is manipulating autophagy differently at different time points of the viral cycle. And specifically, if we look at the very early time points, so the first hours after the virus is entering the cells, there is going to be an activation of the autophagy at those early time points. So we hypothesized that maybe this predictive pathway prediction of increased autophagy could be a reflect of what we are seeing in the cells at those early time points, because this is with the cellular model. So it's only, I'd say, 28 hours of infection. So that was on the cellular part. And we also did a little bit more analysis on the viral protein that were contained in our sEV. So for those of you who are not virologist specialists, I just show here the entry of the HMV virus. So you have the external on the left, on the sham on the left, you have the external envelope of the virus. Inside, you have the geometrical icosahedric capsid. And then inside, you have a little bit of bubbles. Those are not EVs, those are proteins. This is the tegument. And you see that when the virus and the external envelope is going to fuse with the cells, there is on one side the capsid that is going to be the capsid is going to go into the cell that is infected, but there is also all those protein and all the tegument content is going to be released also into the cell. And this is interesting because those proteins and those viral proteins actually have an impact immediately when the virus is fusing and before any viral transcription process. So this is really proteins that are they have an immediate early effect. On the right is the list of all the proteins that we have found in our EVs that are viral proteins. So they are specifically HCMV proteins. They are involved at different time points of the cycle. Some of them, like I said, are really involved in immediate early time points, and some early and some late. This is just the natural cycle life of the HCMV. And what we were really interested in is that we have some proteins that are tegument protein. So the one I just described before. So that would be immediate early 1, immediate early 2, and also PP71, PP65. Were really proteins that will have an impact really early in the natural cycle of HCMV. So we find it was quite interesting to find those enriched in our sEV. There are also some proteins that are involved in different parts and time of the viral cycle. So all the immune evasion process. So HCMV is really manipulating the immune response of the cell host. It's a complex virus. And then later time points, so with the capsid formation and then the virus egress. There are two proteins that I also wanted to highlight. So always viral protein, it's IRS1 and TRS1. So the same thing I was talking about just before, we studying specifically the interaction of HCMV and autophagy. They show that those two proteins are modulating the autophagy in the host cells, so in the natural infection by HCMV. So maybe we can say that there is a potential impact also of those proteins in the manipulation of autophagy that we see just before with the predictive pathway analysis. So when we look at all those data taken together, either the one that come from ex vivo placental culture or the one that come from cytotophoblastic cell culture, we see that we have actually a cellular content, a cellular protein and a viral protein content in the sEV, and that this content could be proviral, so that it could theoretically facilitate further infection. If we imagine that the sEVs are produced by the placenta and then they travel through the directly the blood or the amniotic fluid through the fetus, what is the impact of those sEV that are modified and are carrying viral protein, and they're not carrying virus, they're really carrying those tegumental proteins that we know in a natural viral cycle by HCMV are going to ease and to increase the rate of infection. So to validate this, this hypothesis, I produced some sEV from both the cytotophoblastic model and the placental ex vivo model, and then I put them in contact with fetal cells. So I've been working both with fibroblasts, which are quite classical, fetal fibroblasts, which are quite classical in the study of HCMV virus because they're really sensitive to that virus. And I've also been working with a more relevant cell line when we talk about HCMV infection, which is the null stem cells, because like I said, most of the pejorative outcomes that we will have in the fetal brain and in the neonates after are defects that are in the brain. So here you have different immunofluorescence on the left. So it's the different condition. Every time there is the control with only the infection by HCMV, only the sEV. to assess every time that we don't have any viral particle that were purified and co-purified in our sEV preparation. And then there are the two conditions that are shown up. So the sEV that come from non-infected cells or infected cells put in contact with the cells. So for different times, here I show the time of 2 hours of contact before. And then after 2 hours of contact between the sEV and the cells, a secondary infection by HCMV. And then we quantified the level of infection. So this is the result that are shown here. We did also with different EVs rates. And we showed that there is a difference in the infection rates. So here I'm really talking about secondary infection. So when you put sEV that come from infected cells in contact with fetal fibroblasts, and then when you infect them, you're going to have a higher sensitivity to infection. And this is found both at 50 and 200 EVs per cell. So that was the first part, where we showed that there is an impact of cytotrophoblastic sEV on fibroblast model. That was a little bit of proof of concept, because then we dig a little bit deeper on more, I'd say, relevant models to study this. So then we switched to sEV that were produced from ex vivo placental. So the model I presented before, that was either infected, either non-infected. For this experiment, every placenta has its own control, and it's the non-infected one. So it's really the same patient, the same donor. And then after purifying the sEVs that come from those samples, I put them in contact with neural stem cells. So those cells are quite interesting because they have the capacity to really differentiate into astrocytes and neurons and oligodendrocytes. So this is really what we have at the early time point of embryogenesis in the brain. And we know that the more early the infection by HCMV is during pregnancy, the worse the potential outcomes are going to be. So this is why this model was quite interesting in this context. And then I proceed to the same kind of experiment that I just showed you before. So in infection, putting in contact the sEV that come from infected placenta into neural stem cell, wait a little bit, and then infect. And here we see that we also have the same pattern, which is that the infection, the secondary infection by HCMV, is increased when we have put before the sEV that come from infected cells. So we can conclude that either in reductionist model of cell culture and in more relevant physiologically speaking infected conditions from placenta, we have the sEVs that are produced. They are modified and they are increasing a secondary infection, specifically in fetal cells. So this is a summary of what I've been discussing about for 25 minutes already. Wow. So the first thing that we've done in the team is that we showed that the HCMV is modifying the trophoblastic sEV and also the placental sEV. It is increasing the secretion, and there is a switch toward the proviral protein profile, both in cellular proteins, so Taiwan, for instance, and maybe some more things to explore on the autophagy pathway activation, and also on the viral protein content. So this was interesting for us. This has been shown and published in parallel of our team by another team who've worked on fibroblasts. So, and we do have some similar results. So it's always nice to see that we have concordance data, in my opinion. After this more descriptive part of the project, we did more analysis on the impact of those sEVs. So both if we work in trophoblastic cells or in placental astroculture, and we do have some preliminary results on amniotic fluid, we show that the sEV that come from infected placenta are increasing a secondary infection rate on fetal fibroblasts and neural stem cells. This is the paper I've been talking about. So if you want to have more info on the material and method, or if you want to have a look at the beautiful pictures that I didn't have the time to show you, you can have a look at them. We'll be really happy about it. And I just wanted to finish on a more, say, personal note. I'm a researcher, but I'm also a midwife. So when I joined Cecile Manu Lab, I was really interested in see how we could bridge the gap that sometimes exists between clinicians and researcher. And through a marvelous collaboration with the Toulouse Hospital and the Germatech Biological Center with Nathalie Monard and Melinda Benard, among a lot others, I started to constitute a biobank for congenital infection by HCMV. So the idea was really to collect as many biofluids and biosamples that we could have from the beginning. So really from the diagnosis. Not every woman that is going to have a congenital infection by HCMV is going to have the same care. So the idea was really to, okay, she's going to have an amniocentesis, we can work with that. And then, of course, at the delivery, to collect the cord blood and the placenta. I think that this is important, even if this project specifically has not ended yet and that is still ongoing, the inclusions are ongoing. I think it's quite interesting to have access to this kind of data, because that's the way that we can validate our experimental results with in vivo, in this case, sEV, but other samples also work. And also, that's long-term, but placental sEV has been really developed as potential biomarkers for quite a lot of pregnancy disease, including hypertension and diabetes mellitus. So we could imagine that maybe we'll have also some for HCMV. I would just like to take a few seconds to thank everybody. I think I have 1 minute left, so it should be okay. So I wanted to thank a lot the ISEV for organizing this EVClub, and Kenneth for making it sharp and efficient. Thank you. Also to the FSEV who has supported me through this PhD award. And this is actually the FSEV Congress is the only Congress I went in person before the COVID hit during my PhD. So this is also good memories. Of course, I would like to thank all the people in the team that have participated to this work. So Cecile Manu, my PhD supervisor, a lot of the work has been done together with Hélène Martin. And I'd like to say that my colleague, Shannon Martin, is taking over this project. So very likely in 2 years, either at the FSEV or the ISEV, she will present the new updated data on that project. Of course, we did work with a lot of people and collaborators. So at the Curie Institute, at the Biobank Germatech, all the people that were involved in the transcriptomic and the analysis. And of course, the hospital, the gynecologists, all the midwives, the nurses, the virology team. So it was quite a nice collaborative work. And of course, you for your attention. And I'm ready to take in any questions that pop in the chat or that you might have in person. Thank you.
Thank you so much, Mathilde. And if I'm not mistaken, this picture is also the one that has been awarded during ISEV, during the picture contest, right?
That's exactly that one. I'm reusing this because I think this is the better one that we have. And it's quite nice. I can see, I mean, we don't know if it's EVs, but definitely it was either EVs or debris of EV. And you can see how it's coming like inside this cellular prolongation. So it's placental EV and fetal fibroblasts. Yeah,
that's indeed beautiful. Very nice resolution. So we have Artem that has asked a question in the chat box. Artem, let me first allow participants to unmute. It's done. Artem, I think you can now unmute and ask your question.
Thank you. Very interesting data, really. In the beginning, I was thinking about that some vesicles may contain viral proteins inside or on the surface, and you actually prove it in your proteomics data. But I was curious if there is any morphological difference, like, for example, like size differences between vesicles obtained from healthy cells and infected. Like probably with the through microscopy, you can clearly see the virus itself. But could you see any differences between the vesicles from infected cells and the healthy?
And thanks for your question. No, we didn't show some structural differences between the EVs that come from infected and non-infected either placenta or cytotrophoblastic cells. They are a little bit smaller. They have a content that is different, and I didn't talk about this one, but we also have a difference in the CD63 tetrasphanin. But that is like at the proteic level and not at the structural level. Did I answer your question, Artem?
Yeah, thank you very much.
Thanks.
Awesome. I don't know if Christian, you have questions. I do have a couple in my side.
Well, I mean, yes, I do have one or two questions, but please go first and.
All right. Okay, sounds good. Well, thank you so much, Mathilde, for this very clear presentation. I have some technical questions. I'm pretty sure the data are in the paper, but with respect to this presentation, I think it will make some clarity. So you have mentioned that you performed some RT-qPCRs experiment to be able to actually see if indeed you separated the viral proteins from the extracellular vesicles. And I was wondering if you remember from the top of your head, which genes or which proteins have you checked with qPCR to make this distinction?
Yeah, so what we've done is that we put the sEV that come from infected cells on MRC5 cells. So those are fetal fibroblasts that are super sensitive to HCMV. So our idea was that, okay, we're going to put the EVs on top of the cells. We're going to wait a little bit. And if there is a viral cycle that is induced by that, it means that there is infectious viral particles. So we had two readouts. The first one is the data, so immunofluorescence, so with the immediate early protein. And the other one was a later readout. So I think it's UL55. So that's one of the glycoproteins that are secreted by the virus. And this is one of the pentameric complex that they use for entry.
I see. And jumping on that, you mentioned CD81 was one of the cofactors for the virus entrance into the recipient cells. Have you investigated more this pathway to see how it's going, and if you knock down the CD81, is the infection lower? Do you have any insights?
We didn't did it. It's not that we didn't wanted to, but it's a really good question. HCMV is really a complex virus. Basically, it's a little bit like if you were in a cell and that you were trying to knock down one way of EV egress through the cells. There always one way is going to take over. Mm-hmm. And this is the same for HCMV. If you try to knock down one of the entry receptors, you can have an impact, but it's going to be, there are so many receptors. It's not like if we talk about COVID and ACE2, there's only one. HCMV can use a lot of different integrins. It can use CD81 as a co-receptors. So we thought that if we were going with that, we believe that we have too small impact with only, and I'm coding sEV, to be able to see a difference if we did a KO of that type.
So basically compensation will occur, and okay, you may not see
yeah, exactly. That is also what is happening in clinics for HCMV. There is a lot of different strains and there is a lot of mutation for that virus, but it's in a way, it's an old virus. There is a lot of coagulation between human and CMV, and there is a lot of proteins. It's over 700. Mm-hmm. So it's really, it's a big fat virus in that sense.
Okay, yeah, that's fascinating actually. And do you then think that CD81 plays a role in the autophagy pathways that you describe, or you think it's a bit far away from having this
I
impact?
don't know. This is also something that is quite, in my opinion, really thrilling, is the way that we see, and the more we discover about EVs, the more that we see that we have a lot of similarity between EVs and viruses, right? In the way they are secreted, in the way they are behaving into the cells or in the extracellular matrix and fluids. So I think that we also might not forget that we are infecting the cells at quite a high multiplicity of infections. So there is a lot of virus, and this is also disturbing the cells. I mean, it's not a lytic virus like Zika or other viruses. So the cells are going to survive, but they're going to be a lot disrupted. We can see that they do not have the same phenotype. So in this context, I don't know if the increase in CAD1 that we see is actually having a further impact, or if it's just a reflect of the disturbed cell of origin state.
And how did you actually, this leads me to my next question. How did you establish the rates of extracellular vesicles that you put in vitro? Because you mentioned that you tried 20 EVs, 50 EVs, and 200, if I recall correctly, per cell. Was it based on like some already published data, or like data?
We did not really did it the right way from the beginning. I'd like to say that this was 1.5 years ago. So we didn't have as much literature as we have now in the biodistribution or the EV rates. At our lab, we didn't have any NTA. So we had to go to a collaborator, Everta and Abel, to have, thanks to him, to have this machine. So we were counting with the flow cytometry. So we were a little bit at some point, there were some differences in our counts that we had to put together. So we started with a lot of different concentration of EVs because we were unsure of what was, like you said, biological expected one. And then I didn't show the result, but I also did it with, I think I did it also with like 5 and 2 EV per cells. But this, we don't know what is happening into the placenta for now. So it was really a decision to make a gradient and test different concentrations.
And which one did you trust with the ex vivo experiment after that? Because you showed that afterwards you used the placenta and ex vivo experiment.
We stick with the middle one, so that would be 50 EVs per cell. Because we were also limited by the amount. And I think it's quite interesting when you look at the cells. It's a little bit like when you look at chemicals, right? You expect that if you have the higher the dose you put, the higher the response will be. And we know that this paradigm is not working for the endocrine chemicals, that it's not really the more you put, the more effect you have, but there's going to be a specific plateau where it's working. And I think that it might be the same with the EVs, that we don't know if it's because they're inside or it's just because if they're in contact with the cell. But maybe it's just, if we have too much, then it's going to be overload like the cells. And I think it's really interesting, but that's not what I work specifically on during my PhD. So I cannot give you more insight on that, sorry.
No, that's perfect. I'm going to leave the floor to Christian to ask his questions as well. Thank you so much.
Thank you.
Thank you, Tanina. I have a few questions. Some are about more on the basic side, and some of them are more about the biomedical applications or implications of your data. So talking about this facilitation of a viral infection that you see, I mean, do you have an idea about the mechanism by which this may happen? I mean, is that about the entry of the virus into cells or something else? Do you have some ideas, some hint coming from the proteomic profile that you have?
So specifically, if we look at the proteomic profile, we have the proteins that I talk about of the tegmental proteins. So that would be the proteins that are really involved in the early time points. And we know that if we have a mutant that is positive for those proteins, there's going to be a higher infection. So that has already been known in the field quite a lot. Specifically for this project, I think that it's quite reductive to say that we can only that we look only at the EVs on the proteomic part, right? Because there is also all the non-coding DNA, all the lipids. We have some ongoing work on that, but it's a little bit like if you imagine that even if it's a low quantity, that you have those proteins that are transported by the sEV and that they're coming in the cell that is naive of every infection, those EVs and those proteins, sorry, I meant to say protein, not EVs, those proteins, they're going to prime the cells. They're going to pose the human response of the cell. They're going to really, the CMV is a virus that is literally hijacking the cell, a little bit like any virus, but this one has really a whole manipulative side in the sense that it will really make sure that the cellular environment is is better for him in the sense that he can increase then its further replication. So on the proteomic side, I think that's the data that we have. But like I say, I'm really confident in the fact that we will have some other data. And actually, the other team that has been also working on EVs, I'm sorry, I cannot remember the name of the first author. That's not good, sorry. They also did the similar experiment with fibroblasts and infected by HMV. And they show, even if the purification of sEV was not in the same protocol, they also had an increase in viral proteins. So it seems to be something that is quite replicative and different, that we can replicate in different samples.
Yeah, that's interesting. I have another question that is more about the biomedical implications. I mean, you mentioned at some point that these changes that you see in EVs, sEVs, could be used as a biomarker to assess the presence of viral infection. So, I mean, you have this change, in particular about one surface protein that you can see using the MaxPlex technology. Is that magnitude of change would be translatable into a test that could be used in the context of some sort of clinical research or some sort of studies to bring a test to the market, and whether you think, the question is also whether you think that based on this data you have solid foundations to now move into developing a test, that could be routinely used in that context, detecting viral infection.
There's a lot of things in what you said, Christian. So I think I could actually cut it in three questions, different questions. So the first one is that for HCMV, we do already have some diagnostic markers. So those are not the ones that are lacking in clinics. What we are lacking is prognosis marker. So how is it going to evolve? And in that sense, the data that I presented are really interesting research speaking, but not that much on the clinical point of view, because they are not linked to a specific defect type. They're coming from ex vivo, infected condition.
Yes.
So that leads me to the second part, that I don't think that those proteins specifically in the MaxPlex are the one that we that might be the best candidates to develop markers and biomarkers.
Okay, so thank you very much for the answers to these questions. I guess that we have no other questions from the online audience in the chat.
Yeah, we don't, but I do see Cécile Malnou, former Mathilde PI, in the audience. I'm not sure if she's in front of her screen, but I would like to give her the floor in case she has any comments to add. Proud PI time, I guess.
Hello, everybody. So I really thank Mathilde for a really nice presentation today, as usual, I would say. It's a really nice work from her, and I'm really proud of her work. And as she said, now we have a new student, Charlène Martin, that is now in the team, and I hope this project will continue with Charlène. And I'm really happy to have this nice team with me, and I'm really lucky. And I want also to thank Hélène Martin, also the engineer of the team. This is success of the team, and I want to thank everybody and also our collaborator. And I hope we will continue the story, and we'll be able to give some news in the next ISEV meetings.
Awesome. Well, looking forward to hear more about this exciting study. I don't have any further comments as of today. Christian, feel free to conclude the session.
Okay, so well, I mean, thank you again to Mathilde for a very comprehensive and clear presentation of this work. And also thank you to the participants for the discussion. I think that if there are no other questions, we're gonna end up this EVClub now. And thanks a lot again to everyone. And looking forward to reconvene at some point in some other EVClubs.
Thank you so much.
Unless Kenneth wants to say something before we close this.
No, just thank you very much. It's been wonderful, and it's been a real pleasure to learn more today.
Bye, everyone.
Thank you.
Thank you. Bye-bye, everyone.
Bye.